This is the Physical Activity Researcher Podcast, a podcast for researchers of sedentary behavior, physical activity, and sports. Join for a relaxed dialogue about research design, practicalities, and, well, anything related to research. Learn from your fellow researchers useful and relevant information that does not fit into formal content and limited space of scientific publications. And here is your host, researcher and entrepreneur, Olli Tikkanen.
Welcome everyone. I'm looking forward for today's episode. We are talking about changes in sweat biomolecule levels during exercise. And while this might not at first sound super interesting, it actually is. And we are going to talk also about why is that. Our guest is making her second appearance in this podcast. She's working as a postdoc at Gerontology Research Center at University of Jyväskylä, Finland. She's doing research on skeletal muscle and hormonal aging. Ladies and gentlemen, I'm honored to introduce our guest, Dr. Sira Karvinen. Welcome, Sira.
Thank you. It's good to be back.
Great to have you. So we have been discussing about skeletal muscle and hormonal aging before, and now we are discussing about sweat. So why is it important to study sweat?
Well, in terms of exercise, since that's probably the main topic people are interested when they listen to this podcast, well, sweating is an important part of thermoregulation. And since the evaporation of sweat from skin surface lowers the body temperature. And of course, this is important during exercise as well as during in hot environments. And since sweat is kind of a natural byproduct of exercise, it would be nice if we can find something that we could, use for detecting, for instance, the exercise intensity. Since sweat can be collected non-invasively, so it would be very convenient to collect, for instance, to use instead of blood.
So how did you end up studying this sweat? How did you start this project?
Well, that's a very good question. Actually, the big names behind this sweat project are Professors Seppo Vainio and Urho Kujala. And first, Professor Vainio suggested to harvest sweat samples from athletes. And then Urho Kujala built a team at the University of Jyväskylä. And this project was funded by Business Finland. And it included this collaboration where we planned and harvested sweat during exercise protocols. And the original idea was that the most of the analysis will be done at Oulu University. And so it will be. So later on, we will hear probably more comprehensive analysis from the Oulu University side. But then we came up with our own small side project as we were in this project that focused on extracellular vesicles and microRNAs in sweat. And in our small side project, we used only a very small volume of the sweat, and then also compared that to the serum samples that we had from the same subjects. So basically, it was already a moving train when I hopped in, and I started being a supervisor of the master's student in this project, who was Tero Sievänen. And we ended up having such great results that we thought that why not turn it into a one publication.
Interesting. So you mentioned that one of the funding bodies for this was Business Finland. Could you tell why would Business Finland fund this kind of project?
Well, since I'm not in the board of Business Finland financial things, probably I'm not the best person to answer. And maybe, of course, the professors who applied for the funding would know better. But of course, there's the chance that with our results, the research of non-invasive exercise monitoring systems would take a big leap towards developing actual systems that people could buy and use as they exercise. So I suppose this was the idea behind the whole project.
So practical implications in the study. So how do you actually collect sweat?
I have read and heard of several ways how to collect it, and this can include anything from dripping sweat from the forehead and face into a beaker, or collecting it to a small lab tubes, or even sanitary towels. Um, However, the whole body washdown is considered the most accurate method because there is the sweat runoff from the entire body surface area is collected, and also it does not interfere with normal evaporative sweating. And since some of the listeners might not be familiar with these methods, so when you want to do a whole body washdown of sweat, it involves a construction of a plastic frame that supports basically a large plastic bag within the subject exercises. And then after the exercise is completed, then also the subject and the equipment again is washed with water to harvest all the sweat. But as you can understand, the whole body washdown requires a very well controlled laboratory, a unique laboratory setting, I would say. So therefore, in most studies, sweat is collected from one or more small regions of the body. So the whole body washdown is less used. But when you want to harvest the regional sweat, then of course you again have several techniques. So you can either scrape it or drip it or use filter paper and some kind of arm bags. And there are also commercial sweat collectors available.
Very, very interesting. So did I understand right that in the whole body wash off you put the participant in a plastic bag in a way?
That's how I have understood. I must say I have only read about this. I haven't seen it actually take place, but that's how it was described.
All right, yeah, and if you going for some practical applications, probably this whole body wash off is not one of them. So, what are the limitations and strengths of different methods of collecting sweat?
Well, of course, if you want to do the whole body, if you're capable of doing the whole body washdown, then it's considered like the golden standard. And you get the sweat from all over the body. It doesn't disturb your sweating process whatsoever. But of course, it's probably not the most convenient for the subject. And so it has the limitations, of course, of how many subjects you can take in. And of course, how could you translate that to something that people could use in their everyday life? It's kind of hard to see, at least right now. But then if we talk about the regional sweat collection, that's very practical in many cases. And of course, depending on the method, there is some limitations. And for example, if you cover the skin surface with a collection system that is basically like a plastic thing, it can alter the local environment, and it can affect, of course, the skin temperature when it's put inside a plastic bag, for instance, your arm. So it affects the local area. On the other hand, if you use a kind of open air collection techniques, which would be basically the scraping and dripping, it does allow the evaporation of water from the sweat. So basically, then the sweat that you're harvesting isn't quite the same that you would get if the water is still there with the ratio it's used to.
And which of these methods you are using in your study?
In our study, we used the regional sweat collecting, and since we had bike ergometer for our tests, so the gloves was the most convenient and maybe the most practical way of harvesting the sweat. So we used just basically veterinary gloves that would cover the hands and the arms of the subject. And of course, as the subject spike, so the hands are relatively still there when they bike. So that was for our setup the most practical location to harvest the sweat.
So you are collecting from the arms and hands, and do you just let the sweat to drop on something, or how do you do it? I didn't fully get it.
Yeah, basically, when you put the gloves on the first, subjects, of course, they need to wash their hands rinse with water and with a brush, so that they don't have anything that could contaminate our results. And then you put on the gloves, and then you need to tape them, of course, so that it's kind of it's not open system. So they're basically covered in plastic bags, the hands and arms, and then the sweat just drips. So basically, in the end, we just spin the gloves. The sweat will be at the very fingertips of the glove, and then we can pipette them out from there.
All right, interesting. And how is it with different location where you harvest the sweat? Is it different in its constitution from different locations?
Well, unfortunately, it is. So our results might be different if we use some other location. And the most known differences are that there are basically two different kinds of sweat glands. So there are ecrine and apocrine glands. And these ecrine glands occur over most of your body, and they open directly onto the surface of the skin. And also the ecrine sweat glands are the most numerous, and they are responsible for the highest volume of sweat that you produce during exercise or in hot environments. Whereas the apocrine glands, they open into the hair follicle actually, and that leads to the surface of the skin. So basically those are the glands that you have in your armpits, for instance. So it is well documented that many sweat micronutrients, for instance, vary depending on the anatomical site where it's harvested from. And then, of course, depending on the area, there might be something in the skin surface that contaminates or impacts the sweat sample that you harvest. So there are differences.
Yeah.
This podcast is sponsored by Fibion, a research device that has been shown to be valid in tracking sitting, standing, physical activity and energy expenditure. Get scientific validation and learn more about Fibion at fibion.com/research. Fibion, from researchers to researchers.
And probably if you think of practical applications, probably a wrist-worn device could be a good solution, as you have already smartwatches and different running computers or whatever you would like to call them. So, and you are collecting from the wrist. How do you see? Could you get better data from other locations, or do you think that the wrist is a good location?
To be honest, I really don't know. I don't think we have a comprehensive data of, comparing different locations in terms of exercise and what kind of response you get. But if we think of, for instance, the problem is normally that you don't have sufficient amount of sweat because there's not much analytes. So in that sense, I'm not sure if wrist would be the best, whether people sweat enough from that area. So I would think that's my first concern. And then the second one would be that what are the results if we take it from wrist, or if we take it from, for instance, forehead, or something that a runner or a skier or whoever could actually wear something there.
And when you analyze, how much do you basically need with current technologies? How much do you need sweat to be able to analyze it accurately?
Well, it depends a lot what you're looking at. And for instance, many of the previous sweat studies have concentrated on the proteomics or the metabolomics of sweat. And then you need tens of milliliters. And normally that means also that you need to pool the samples. You don't get enough from one study subject, so you probably need to pool the sweat. And of course, that's not a good thing if we want to measure a single person. But for instance, as we started our study, we wanted to check the microRNAs, since the good thing, or there are many good things, they have been studied a lot in relation to exercise. And then also, since you can measure those with PCR, so then basically the method that we use is to amplify the amount of the analytes that we have there. So with our method, since it's based on amplifying the signal, basically, we could use only 1 ml of sweat. So that's a very small amount of sweat that we used in our study.
And so when you actually analyze sweat, what does it actually consist of?
Well, sweat is, of course, it's mainly water, but then it also contains electrolytes such as sodium and chloride and other micronutrients, for instance, calcium, magnesium, some vitamins as well. And then the metabolites that have been studied previously, such as glucose and lactate, which is interesting in terms of exercise. Of course, there's amino acids, and there's also other compounds with very small amounts, such as cytokines and cortisol. So there's many analytes to look at. It's just that there's with such small amounts that it needs a large amount of sweat, and then a very accurate methods to actually get a signal from there.
And what do you see as the most interesting things to look at?
Well, there could be many things. So far, the literature thus far hasn't found very promising, for instance, metabolites that would, reflect the intensity of the exercise, for instance. So it hasn't shown to be very accurate. And then also lactate would be interesting in terms of exercise, since most of the listeners probably know that you measure the lactate from the blood with, for instance, in competitive runners or cyclists or whoever does exercise in high intensity to design the exercise training regimen and the race pace, for instance. So it would be very practical if we could, for instance, measure the lactate from sweat. However, sweat lactate concentration generally does not correlate well with that of the blood. And at least to some part, apparently, it's due to that the sweat gland itself also produces lactate. So there's a lot of interference going on there. So hence, for instance, what we studied were the microRNAs. And at least to us, our results were quite promising. However, in terms of talking about future non-invasive monitoring systems, I am not sure if we can find out a way to detect those robustly from sweat. So there are many things that we can study, but there hasn't been like a great success with any of these. But hopefully we'll get there.
And why did you go and start studying the microRNA in your exercise study setup? What was the reasoning behind this?
First off, for those who are not familiar with microRNAs, so those are a small non-coding RNA molecules. The function of those microRNAs is that they bind to its target messenger RNA, and hence they block the protein translation. So it alters the gene expression in a post-transcriptional level, and that's the function of those. MicroRNAs, in terms of exercise, have been widely studied, but in plasma. So there are several microRNAs that give a response to exercise stimulus, and especially in endurance exercise stimulus. And hence those are kind of promising biomarkers, at least in plasma, of the exercise. And then also microRNAs, because they're short, so they're very promising if we talk about acute exercise responses, because they're fast to produce. And then cells either secrete them into the bloodstream to communicate with other tissues, and then they're also actively packed into these vesicles and then transported to target tissues. And microRNAs have several functions. And of the microRNAs that we studied here in our study were well-known exercise-responsive microRNAs that have the function, for instance, in inflammation or hypoxic adaptations. And many of them were mediators of angiogenesis. So since they were very well documented in serum or plasma already to respond to endurance exercise, these were the ones that we handpicked kind of based on previous literature and see whether we can find them in sweat and whether we could see some kind of response also in sweat.
And what do you see as the most promising markers in general, also in plasma from mRNA?
Well, that's a kind of tricky question. There's so many. If you ask in terms of plasma or?
Yeah, like in general, you said that some of them respond to inflammation, some to hypoxia. What do you see from practical point of view that what are the most interesting or promising biomarkers?
Well, that's a tough question because it's many to choose from. MicroRNA 21 is probably the one most studied one because it also responds to various kind of stresses. So it doesn't have to be exercise. It can be just some kind of stress due to the body. Whereas microRNA 146 is probably the most cited in terms of exercise, and especially in endurance exercise. And it also both of those are involved in inflammation and hypoxic adaptations. So those would be the two that you most commonly find in literature. And also the microRNA 21 is kind of fun for the reason that basically you can find it anywhere. So if you just want to check if there are microRNAs, normally we check the microRNA 21. But of course, then it can respond to various stressors. So it would be nice to find a microRNA that is more specific to certain kind of exercise stimulus, for instance. So in that sense, I'm tempted to say, for instance, microRNA 146 for now, at least.
All right. And in your study, when you were analyzing the sweat, what were your main findings?
Well, we were the first to show that the sweat extracellular vesicles contain microRNAs, since no one had studied this before. So that was kind of cool finding, first of all. And we were actually able to find all of the microRNAs that we had in our study in sweat extracellular vesicles. And also, we found that 2 of the 6 microRNAs that we had handpicked responded at least to one of the exercise regimen that we had. And of these microRNAs, the microRNA 26 was actually, that was very abundant also in sweat. So it increased in sweat vesicles as the exercise intensity increased. So I think that's the most promising finding that we had from sweat.
So the mRNA 26 increased with the intensity. You mean like when the heart rate goes up, or the lactate, or which part of the exercise intensity it kind of responds or relates to?
Well, it depends probably, it might depend also at the study subject, but going from, since we had basically 4 different or 3 different exercise regimen, and then we had the sauna control. So here I'm referring to that they have the anaerobic threshold test, which was done at the aerobic threshold, and then anaerobic threshold test, and then the VO2 max, which is of course all out kind of test. So moving on from the aerobic to anaerobic to VO2 max, it increases.
Interesting. So how do you see the practical applications? Do you see this could be used somehow to measure the intensity of the exercise?
Well, it does look promising, at least. Of course, our study was done only with 8 subjects, so it has its limitations. However, already with the N of 8, we were able to have significant results. So it's kind of a limitation, and then again, it's kind of a very promising that we can see it already with such a small N. So I would call this like a preliminary data or a preliminary study. So first we should check this with larger population if we can see the same finding. And then probably we could go on digging more into whether this microRNA 26 actually is something to look for to in future, and whether it's worth to start developing systems that could actually detect.
And now, as you are looking for the microRNAs, how difficult is it to analyze them? Like if we think of a wearable in the future, how do you see this? When could this kind of thing be ready?
It's very difficult to say. Now the system that we have been using is basically, it's a very commonly used lab technique, but of course it's something that doesn't take place in your watch, for instance. So you would need something else, a detector that binds to it and then gives some kind of signal that the microRNA exists in the sample. And I'm not sure how easy that is to develop. However, techniques take leaps forward, I don't know whether it's every year, but probably something that can be solved. But I'm not probably the person who is going to do that. So I'm going to stick with the research or the basic research, and then someone else can maybe put their brains and mind into this, developing wearable sensors.
Yeah, I understand that. So you said that your paper was a small part, and they are doing the bigger part in Oulu. Could you tell more about this bigger project? What are they looking from sweat?
Well, they will have more comprehensive analysis since, of course, from the arms you can harvest several milliliters of sweat, and we saved only a small portion for us. And so far we have used only the 1 milliliter of that. So they will have a lot more to work with. So what we're looking forward is to hear something about the protein profiling, for instance, from the sweat, but we just don't have it quite yet. So we're very much looking forward to their analysis. And I'm not sure how much I can tell about it.
Yeah, I understand. So what do you see as the biggest challenges when you are studying sweat?
Well, of course, first, how do you harvest it? Of course, there are already the wearable sensors that have some kind of capillary tube or filter paper. So basically, you can harvest sweat. But then it comes to the small amount of the analytes. So, for instance, if you want to study the protein profile of sweat, then you need several mLs or tens of mLs of sweat. So that's not very convenient for the user who's using it. So I think it's mainly for the analyzing the analytes. So it needs to detect very small amount of analytes and in a robust way. So I think those are the main challenges right now.
Mm. And now your study is about exercise part. Do you see some other applications for analyzing sweat, for example, in relation to health and disease or something else?
Well, yes, sweat is currently, for instance, used, for example, the diagnosis of cystic fibrosis. And to my understanding, that is currently the only like health or disease related method that sweat is used for. But for the sweat test of the cystic fibrosis, it measures the amount of chloride, so which is a part of the salt in sweat. And it's actually the golden standard for the diagnosis right now. So it is used for one disease so far, but I don't know whether it could use for other diseases as well. And of course, well, in sweat, certain drugs, if you use them, they accumulate into sweat, as well as ethanol, for instance. So it could be used something else for as well.
Yeah, interesting. So how do you see the future of sweat-based analytics in exercise and also in other fields?
Well, in terms of exercise, for instance, since that's what we studied here, if we want to use the sweat as substitute of serum or blood sampling, so we need to bear in mind that, for instance, what we found was that extracellular vesicle population in sweat is different from that of serum. And as I said, for instance, the lactate production of the sweat gland itself, it makes it difficult for it to be as a substitute of serum. And of course, when we saw that 2 out of the 6 microRNAs studied in our project gave actually a response to exercise in sweat, it is kind of a promising signal. And but the problems really in the future would be how you collect the sweat and where you collect it from, and of course the fact that can we and when we can detect so small amount of the analytes. And of course, what is the target? Will it be the microRNA? Is that the most promising thing so far, or could we actually get some metabolite or something else, or a protein or something that could be more interesting and maybe easier to detect from sweat? So hopefully we will have answers to these when we get the more comprehensive analysis.
Yeah. And if I go a little bit back, you said that some of the microRNAs are responsive to inflammation. So do you see a response already during exercise in the inflammation, or how does it go?
Well, it depends on the person probably and the exercise intensity, but of course, exercise is known to cause mild inflammatory responsive markers as well. So it kind of makes sense that also the inflammatory or inflammation related microRNAs respond to exercise. But it probably depends also on the person and what they have done before and how the body responds to exercise and how intense that is. So it's probably hard to say whether everybody responds the same.
But anyway, for some people it can be so fast that already during exercise you can see it you can see the inflammation response in the microRNA. Did I understand it correctly?
Well, at least in the microRNA side, like I said, the microRNAs are very fast response. So they're kind of the very beginning of certain signaling routes. So at least you can see the very beginning of that signaling route. But of course, then we need to bear in mind that these are kind of the functions of the microRNAs that we know so far. And microRNAs are not gene specific. So one microRNA can bind to several messenger RNA. So what we say inflammation here might be something else in the body, since we don't know yet everything it controls. So this is just our educated guess of what is it doing in the body when we see a response.
Do you plan to have other protocols? Now you have the endurance exercise. I'm thinking like, for example, sledge counter movement jumps, which would definitely create breakdown of muscle tissue or some micro damage, which would be inflammation. So maybe with this you could check that is it probably related to inflammation or muscle damage, micro damage?
Well, that's a good idea. And to be honest, we had some of the study subjects doing leg press for sweating. And what I heard, it was quite awful for them because it's very heavy, you know.
Yeah.
And to make it so much that you can actually harvest sweat. But unfortunately, we had so small n that we couldn't run analysis from that. But yeah, we could definitely go into those samples and, you know, take a look.
Yeah. And I think it could be because, for example, in strength training, if you're looking for hypertrophy, your kind of idea is to cause micro damage in the muscle, and one of the stimuli for the muscle growth is inflammation. So maybe you could create a wearable device for the strength training people, and right now they don't have too many wearables that would actually work for them because they are mainly about aerobic exercise.
Yes, you're right. And, you know, it could be done.
Yeah. So maybe the strength training people are getting excited of the wearable coming so they can see how much they are producing micro damage in their muscles. So it's been interesting discussion. Is there something else you would like to add into this discussion?
I don't think so. I think we have covered it quite well.
Yeah, I agree. And you're doing very interesting research. Is there some kind of collaboration you are looking for? Is there some kind of institute you would like to collaborate within this project?
Well, I'm always looking for collaboration. I'm not sure, though, with the world situation right now, how well it could happen. But for instance, we still have some of the sweat samples left. So if someone is interested, if you have a great idea of what to look for, and you would be interested to collaborate in analyzing it, we'd be happy to discuss.
Yeah, very, very interesting things. So thank you, Sira, for taking the time for this podcast episode.
No problem. It was my pleasure.
Thanks for joining us this week on Physical Activity Researcher Podcast. If you like the show, make sure you never miss an episode by subscribing or following the show on Twitter. This podcast is made possible by listeners like you. Thank you for your support. If you found value in the show, we would really appreciate a rating on Apple Podcasts or whichever app you're using. Or if you would, in a real old school way, simply tell a friend about the show. It would be a great help for us. We have a fantastic lineup of guests for forthcoming episodes, so be sure to tune in. Thank you all for your support and have a great day.